Organs of the immune system, secondary lymphoid organs. In the intricate and vast network of immune system is present the secondary lymphoid organs, which help in eliciting an immune response. In this session, let us see and how do these secondary lymphoid organs function and which are these secondary lymphoid organs. So let us look at the learning outcomes of this session. The immune system has an integrated lymphatic system which enables lymph to be collected and returned to the blood, thereby also carrying lymphocytes from tissues. So basically, the lymphatic system is able to carry a lot of lymphocytes from one place to another place. The secondary lymphoid organs are distributed in the body and are associated with the lymphatic system. Most lymphoid organs have germinal centers and follicle cells, which, on getting challenged by an antigen, lead to giving rise to cells and antibodies that can be specific in its response. These are responsible for both non-specific and specific immune response. Now let us look at the lymphatic system per se. One can say that this is a parallel system to the circulatory system. And in fact, what is interesting is that you would find that at various places, you will have the secondary lymphoid organs present, which are, all on the surfaces. That means the lymphatic vessels are very close to the secondary lymphoid organs. Another interesting facet that is there is that just like the circulatory system, you have lymphatic capillaries which converge to form the lymphatic vessels, and these lymphatic vessels then converge to a larger vessel that is able to return the lymph to the blood itself. Now the question that comes up is, how is the lymph formed? So, what has been observed is that as blood circulates in the circulatory system through the capillaries, the plasma or the clear liquid without the cells are able to seep into the tissues that are surrounding these capillaries. So, these, fluid that has escaped from the blood into the surrounding tissues, okay, generally would get back into the capillaries again. That means the blood vessels again. However, some amount of fluid remains within the tissues, and these fluid is what is also called as the interstitial fluid. So this interstitial fluid is what is called as lymph. Now that lymph is taken up by or collected by the lymphatic capillaries. So what is in green, as you can see, they are the lymphatic capillaries, and interestingly the lymph vessels have what is called as inbuilt valves. They're called as lymphatic valves, and these valves ensure that the lymph moves in one direction. So all lymph that is collected from all over the body eventually comes to a thoracic duct. Okay, this is the collecting duct in which all the lymph comes through, and that lymph is then you know put into the blood vessel or the blood vein again. So it basically releases all the lymph again to the circulatory system. Now, all along you will find at various regions, these vessels are impregnated with what is called as lymph nodes. You can see over here, there are certain nodes that are formed all over. So these are what are called as lymph nodes. And these lymph nodes are actually secondary lymphoid organs. You can see over here how spleen is present just in the upper abdominal cavity. You have the tonsils which are present over here. So you can find several different secondary lymphoid organs. You will have in the gut also the secondary lymphoid organs present. So now, when an antigen enters tissues, okay, so these antigens can flow into the lymphatic vessels along with the lymph, okay, And once the lymph with the antigen flows and enters lymph nodes, then in the lymph nodes, the antigens get trapped. That is because of the structure that lymph node has, the antigens get trapped over there. Now, that is a strategical thing that is happening. The lymphocytes also that are there in the tissues will be collected again by the lymphatic system, and the lymphocytes will be also carried along with the antigen all along the lymphatic vessels. Now, these lymphocytes, therefore, can encounter the antigens that have been trapped in the lymph node, and in response to that antigen, the lymphocytes get activated. And due to their activation, there is an immune response. Organized tissues, that is, organized lymphoid tissues, are located along the vessels of the lymphatic system, as just mentioned. Now, you can see over here how this in blue, over all around in the blue, is the tissue space, and you can see how the lymphatic capillaries will take in the lymph, and from the lymphatic capillaries you will find them moving into lymphatic vessels. From lymphatic vessels they will get into what is called as the afferent lymphatic vessel, and these afferent lymphatic vessel end up in what is called as a lymph node. Once they enter the lymph node, from the lymph node, again you have an efferent lymphatic vessel coming out. And so you can see how the lymph that has been collected from the tissue goes right into the lymph node, and from the lymph node again back to a lymphatic vessel. So the point that is to be noted over here is any antigen that is taken from the tissue space will land up in the lymph node. And this entire system has what is called as lymphoid follicles. The lymphoid follicles can be differentiated into primary follicles and secondary lymphoid follicles. The primary follicles, they basically comprise what is called as dendritic cells. These are immune cells and resting B cells. Now, once you have this getting challenged by an antigen, okay, then the lymphocytes that are present over here, which are resting, they get activated and begin proliferating. So when they reach the secondary follicles, so from the primary follicle the lymphocytes enter the lymph node. Within the lymph node there are several germinal centers. Inside the germinal center you will have non-dividing B cells, you will have T helper cells and macrophages or dendritic cells. So all these together will activate B cells, and the B cells will begin proliferating. So you will find that the secondary follicle has a lot of proliferating B cell. And what is also interesting to note is that in this germinal cells, there is a phenomenon that is called as B cell selection happening. What do we understand by B cell selection? By B cell selection, what we understand is that as B cells divide, okay, in the process of dividing, there may be a lot of mutations that are also happening, and because of these mutations, every B cell will be a little different. What has been observed is because of the different B cells that are formed, and please note that B cells have on its surface antibodies. So you will have different antibodies with a specificity to bind to a particular antigen. Now, therefore, the understanding is that if there are 100 B cells formed, out of the 100 B cells, about 80 B cells that are formed contain antibodies that are not specific for the antigen that the primary follicle was challenged with. So the remaining 10 or the remaining 20 that are there, they have more specificity to bind to the antigen, and so those will survive or those will be sustained, but the remaining 80 will undergo degradation by apoptosis or by some other process, or even the fact that there are macrophages present, the macrophages can just simply engulf them. So therefore, B cell selection is basically trying to ensure that only those B cells proliferate that are specific to the antigen that the lymphatic system has encountered. So this is very important with respect to secondary lymphoid organs. You see, the secondary lymphoid organs will specifically produce an immune response against a specific antigen. And that happens when they actually encounter an antigen. So this is exactly what happened over here. Now, which are the secondary lymphoid organs? So, of course, the secondary lymphoid organs are spleen, lymph nodes, Peyer's patches, adenoids, tonsils, and MALT. In fact, Peyer's patches, adenoids, and tonsils are actually part of MALT itself. What is MALT? MALT is mucosal lining associated lymphatic tissue. And as mentioned earlier, this is a response, or these generally give a response only after they have encountered or they have been exposed to an antigen. And therefore, many of these give rise to what is called as adaptive immune system. So let us now look at these different secondary lymphoid organs and understand what they are doing. Before going into that, it is important to know that spleen and lymph nodes are the most well defined secondary lymphoid organs of all the others. We first begin with lymph nodes. So the lymph nodes basically have a structure which looks like an encapsulated bean shaped structure, something like the kidney as well. And it has clusters or at junctions of lymphatic vessels. Okay, so they are basically, like mentioned earlier, they are present at junctions between several lymphatic vessels, and you will find that many of the afferent lymphatic vessels enter the lymph node, so they pour in all the lymph into the lymph node per se, and you will find that the lymph actually moves from one region to the other in the lymph node. Now, the lymph node is generally subdivided into cortex, paracortex, and the medulla. So, the yellow is what is called as cortex, the orange part is what is the paracortex, and the green part is what is called as the medulla. Each of these, whether it is cortex, whether it is paracortex, or whether it is medulla, all of these would have certain specific microenvironments. And these specific microenvironments are very important for contributing to the proliferation of B cells, for the response, for the immune response to be generated. As mentioned, the lymph will come from the afferent, lymphatic vessels, will go to the cortex, then to the medulla, sorry, then to the paracortex, and then to the medulla. So this is how the lymph will be traveling. Okay, it would reach the medulla at the end. Now, the cortex generally comprises the germinal centers. As mentioned earlier, germinal centers have what is called as a non-dividing B cells. It has the T helper cells. It has the follicular dendritic cells, and it also has macrophages. And together, these are able to, kind of activate the primary follicle. Now, the paracortex region has more of the T helper cells, and these T helper cells, along with digitating dendritic cells, are able to activate lymphocytes. So, they are able to activate generally B cells. So, the medulla, which is the last part of the lymph node, will therefore contain B cells that are already proliferating and producing antibodies. But one has to remember that the cortex per se that has the germinal center along with the primary and secondary follicles are actually challenged or are exposed to an antigen, and in response to that antigen, in the paracortex, the T helper cells and the dendritic cells process the antigen, and because they have processed the antigen, based on the antigen processed, you would have specific B cells proliferating and producing antibodies that can bind to the antigen that in the first place these cells had, or these um, regions had encountered. So from the efferent lymphatic vessel, therefore a large number of lymphocytes would move out. and this would this would be more than the number that had entered the node. So you can basically look at the fact that the lymphatic vessels do collect lymphocytes from the surrounding tissue, but when you have the lymph coming in and at the medulla region you have more and more B cells dividing and forming more of the effector cells, then those will also come out from here. So the number of lymphocytes that entered at this point would be much lesser than the number of lymphocytes that are leaving the lymph node. So you can see that there is an immune response. There is a heightened production of B cells against the antigen. Now let us come to the next secondary lymphoid organ, which is the spleen. The spleen helps in immune response to antigens that are present in blood. So lymph looked at antigens that are present in the tissue spaces, okay, while the spleen is looking at an immune response to antigens that come from the blood or are present in the blood. Now, the spleen per se is again a bean-shaped structure, and you can see that this is present basically in the higher abdominal area, and you have a splenic artery that is there and a splenic vein that is present. So, in the spleen, what happens basically is that the blood is filtered. Now, as blood gets filtered, if there is antigen present in the blood, then the antigen gets trapped in the spleen. And the lymphocytes also which are present in the blood, when the blood is getting filtered, the lymphocytes also pass through the spleen. So the basic idea is that when the lymphocytes pass through the spleen, they are able to get exposed to the trapped antigen, which is what is important. If the lymphocytes can see the antigen or bind to the antigen or interact with the antigen, then an immune response can be generated. So you can see both lymph node as well as the spleen are ensuring that the lymphocytes somehow are able to encounter the antigen. Now, when you look at the cross section of the spleen, you will again find that you have a capsule and you have several, um, dividing regions which are called as the trabeculae. So you have what is called as compartmentalization within the spleen itself. And you would interestingly find that this becomes the outer red pulp, okay, and this becomes what is called as the white pulp. So in the spleen, you definitely have the red pulp and the white pulp. The red pulp, what it does or what happens in the red pulp is the old and the defective RBCs are destroyed. Even if there is hemoglobin, etc., that too gets degraded in the red pulp. And the macrophages are present in the red pulp. They, in fact, are responsible for helping to engulf the old and defective RBCs. So they are getting degraded thanks to the macrophages that are present. The white pulp again has primary follicle, as you can see. It has another zone which is called as the marginal zone, and then it has a third region which is called as the periarteriolar lymphoid sheath, or the PALS. This all these three together form what is called as the white pulp. Now, the primary follicle is rich in B cells. The PLS actually comprises lymphocytes, or rather the white pulp comprises the lymphocytes. So just mentioned, primary follicle is rich in B cells, PLS is rich in T cells. And these T cells are again specific T cells with a specific T cell receptor present on them. Now, blood that carries antigens, when it reaches the marginal zone, okay, you would have the antigen getting trapped there. And this marginal zone has several interdigitating dendritic cells that processes these antigens. And having processed the antigens, the dendritic cells activate the T helper cells to in turn activate the B cells, and the B cells start proliferating. And once these B cells are starting to proliferate, they differentiate into what is called as plasma cells, and the plasma cells produce antibodies. Now, the antibodies that are produced by these plasma cells will be very specific for the antigen that was trapped in the marginal zone in the first place. So that is the beauty of the secondary lymphoid organs. That is, they will produce an immune response specifically for an antigen that they have encountered. Now we come to the next set of lymphoid tissue, which is mucosal associated lymphoid tissues. So this actually comprises several lymphoid organs, and they are all associated with mucosal linings. So you could have it in digestive system, because we know digestive system has a mucosal lining. You could have it in the respiratory system, and you can also have it as in the urogenital system. So digestive system, respiratory system, and urogenital system are major points of entry of antigens. By breathing in air, okay, you are able to take up a lot of pathogens from outside. By eating food, in the food, you could have foodborne microbes, or you could have foodborne antigens that come in. Similarly, through urogenital system as well. So some MALT organs, that is lymphoid organs, are diffused. That is, they don't have a very particular structure. They are just diffused lymphoid organs, while some are highly structured. Of this highly structured MALT secondary lymphoid organs are tonsils, adenoids, and Peyer's patches. And all of these are generally associated with plasma cells, and we all know that plasma cells are the ones that are producing or secreting antibodies. So, effectively, like the lymph nodes and the spleen, even MALT is eventually responsible for activating B lymphocytes to differentiate into plasma cells that give rise to antibodies. And these antibodies are specific to antigens that have entered through one of these systems. And interestingly, what has been observed is the number of the plasma cells that are formed exceeds that which is formed in the lymph node, spleen, and bone marrow all put together. So therefore, MALT is definitely a very active system in terms of production of antibodies. Let us now look at tonsils, which is one of the MALT lymphoid organs. So tonsils is present three places. One is lingual tonsil, which is just behind the tongue. One is the palatine tonsil, that is sides of the upper part of the tongue per se, and you have another which is called as the adenoid. This is more closer to the nasopharyngeal cavity that is there. Now when one takes a cross section of the tonsils per se, again what you would observe is and it will encounter antigens that enter through the nasal and the oral route. So all the three, whether it is the palatine tonsil, or whether it is the lingual tonsil, or whether it is the pharyngeal tonsil, all of them basically are a meshwork of reticular cells and fibers that hold macrophages, lymphocytes, granulocytes, and also mast cells. So therefore, whether it is lingual tonsils or whether it is the palatine tonsil, you can see over here the follicles that are there, and these follicles will in turn have germinal centers, and these germinal centers, once they are encountering an antigen, they will be able to activate lymphocytes, and these lymphocytes will then give rise to an immune response. So this is what was mentioned. Now we come to Peyer's patches. Peyer's patches is found in several places in the gut itself and even in the urogenital system. Now, in fact, Peyer's patches has been studied a lot. This is a cross section of the intestine where Peyer's patches are found. As you can see over here, we know that the the outer mucosal lining is just a simple layer of epithelial cells, but they are convoluted to form what is called as the villi. Now, we have within this a region which is called as inductive site. So what is an inductive site? Inductive site is a site from where the antigen can enter. So from the intestinal lumen, you can have the antigen actually enter the epithelial or the entire gut lining, mucosal lining. So the outer mucosal epithelial linings, they contain what is called as intraepithelial lymphocytes, IELs, and these intraepithelial lymphocytes have been found to be specialized T cells. Then just below the epithelium, you have what is called as a lamina propria. And this lamina propria contains B cells, plasma cells, T helper cells, macrophages, etc. So evidently, when you have the antigen coming in, it would be encountering B cells, plasma cells, T helper cells, etc. The submucosal layer has what is called as the Peyer's patch, and this Peyer's patch comprises the primary follicles, the secondary follicles, the germinal centers. All of these are basically present over here. These, on encountering the antigen, will produce an immune response depending on the way the antigen is being processed. Now, at the inductive site are present certain specialized cells which are called as M cells. And these M cells are able to basically allow the antigen to enter through the mucosal lining. Now, when you look at the M cells more, more close, you will find that these M cells, which are also part of the epithelial lining, are not same as the epithelial cells that are present. These epithelial cells are all ciliated. So you have the villi that is present, but the M cells do not really have a defined villi. However, they are able to take in antigen, so they are able to endocytose antigens. And interestingly, the M cells also have a very deep invagination. So you can see this is a very deep invagination at the basolateral region of the epithelial cell. So this is basically a pocket, and the pocket is containing B cells, T helper cells, macrophages. All of them are present over here in certain amounts. Now let us look at uh, what actually you see. Okay, so you find, we mentioned that within the epithelial lining you will find the IELs, that is intraepithelial lymphocytes. So these are the intraepithelial lymphocytes that are present, and these are generally CD8 plus T cells. So they are T cells per se. Just below the M cell, okay, at the inductive site is present in the lamina propria is present an organized lymphoid follicle. So in this organized lymphoid follicle, when the antigen enters, you have the antigen getting processed, and having the antigen getting processed, the B cells get activated, the B cells get differentiated into plasma cells, and the plasma cells are responsible for secreting specifically IgA. So this IgA is actually a secretory antibodies. You will find that many mucosal linings, at many mucosal linings, you have IgA being secreted. Now we come to another lymphoid tissue, which is the cutaneous associated lymphoid tissue. So this is just below the skin per se. So skin epidermis too has been found to have the intraepithelial lymphocytes, which are T cells again, and they have very limited specificity. So they do not have too much of specificity to bind to the antigen. This epithelial lining has certain specialized cells called as keratinocytes. These keratinocytes are able to secrete cytokines. Cytokines are growth factors which can induce cell division. And these keratinocytes are also able to process antigens. So you can see that the cytokines and the processing of the antigens are responsible for activating lymphocytes further. The epidermis also has Langerhans cells, which are dendritic cells. So these Langerhans cells are able to engulf antigens. So in fact, they are dendritic cells which are not interdigitating. So they can engulf the antigens, and from having engulfed the antigens, they can move on to the lymph nodes, and there they become interdigitating dendritic cells. Now, as interdigitating dendritic cells, they are able to process the antigen, And having processed the antigen, they activate the T helper cells, and the T helper cells in turn will activate the B cells, and those B cells get differentiated into plasma cells, and plasma cells will produce the antibodies. Now, just to look at how the lymphoid organs may have evolved. So only across vertebrates, basically, when one observes, lamprey, this is part of the Adnata family in the vertebrates. As you go to trout, this is a type of fish, then frog, chicken, and mouse, you will find that each of these has GALT, that is gut-associated lymphoid tissue. GALT, that means the secondary lymphoid tissue gives rise to non-specific immunity. You will not find adaptive immunity associated with lamprey. But when you move on to the mice, you will find that the mice not just has the galt, it has the it has the thymus, it has the bone marrow, it has the spleen, it has the Peyer's patch, it has the lymph nodes, it has everything that is required for both non-specific as well as specific immune system. So therefore, whether it is frog, whether it is chicken, or whether it is mouse, you will find that they have slowly, gradually developed bone marrow and thymus, which is required for adaptive immunity. So that is something that is interesting to be observed, that within the vertebrates itself, you can find that lower vertebrates don't have, that well defined an immune system as compared to a higher vertebrate. Let us make the conclusions, therefore. Lymphatic system carries lymphocytes and enables trapping antigens entering through the different ports. The lymph that arises from the blood finally enters the blood again, and in the process, it is carrying lymphocytes. And as it carries the lymphocytes, it can get exposed to antigens as well. Spleen and lymph node are highly defined lymphoid organs in which the trapped antigens are processed to activate lymphocytes to give specific immune response. MALT comprises lymphoid tissues or organs found in the digestive tract, respiratory tract, as well as urinogenital tract. Tonsils, adenoids, Peyer's patches are MALT that are well understood and are involved in mucosal immunity, which is generally a non-specific response. Several lymphoid organs have evolved in vertebrates that enable both non-specific and specific immune response. Secondary lymphoid organs, therefore, distributed in different parts connected to the lymphatic system, contributes to processing antigens to elicit an immune response. Thank you.